Hello everyone, welcome to my channel. Today in this video I am going to discuss about structure of chromosome. So for the better understanding of cell cycle and cell division, before that we need to understand about the chromosome. Now what is chromosome? Chromosome is a genetic material which is present in the cell. Structure of chromosome, what I am going to discuss. First of all, I'll discuss about what is chromosome, where it is present, discovery of chromosomes, components of chromosomes, structure of chromosomes, and what are genes. First of all, I'll discuss about what is chromosome. A human body is made up of millions or trillions of cells. So in each cell there will be a nucleus, and within this nucleus genetic material will be present. Thread-like structure will be present inside the nucleus, that is called, if we stain with a suitable dye and see the cell under the microscope, we can see the nucleus, and within the nucleus contains most of the cell's DNA, which is organized into discrete unit called chromosome. Each chromosome contains one long DNA. Each chromosomes contain long DNA molecule associated with many proteins. So we'll see the definition of chromosome. Chromosomes are highly coiled ribbon-like structure formed by the condensation of chromatin fiber during cell division. See, this is the structure of chromosome. So if we talk about the number of chromosomes present in humans is 23 pairs of chromosomes will be present in the humans. 23 pairs of chromosomes are present in humans. Definition: chromosomes are highly coiled. Chromosomes are highly coiled ribbon-like structure formed by the condensation of chromatin fibers during cell division. So what happens in a cell? There is a nucleus, and this nucleus contains chromatin material. This chromatin material which contains information, genetic information. This genetic material found in every cell, and it stores all the information about the organism, and that can be inherited, means it can be transferred from one generation to the another generation. So these chromosomes are present in each and every cells, and most of the DNAs are present in the nucleus. So chromosomes, the structure of chromosome will be, the structure of chromosomes will be like this. The structure of chromosome will be like this. When it get condensed and coiled, it forms a ribbon like structure. When this kind of structure is formed, this kind of structure is formed only during the cell division. The chromosomes get condensed and form this kind of structure when the cell is in the at the time of cell division. But when the cell is not dividing, the chromosomes are present in a thread-like structure present in the nucleus. See, this is a cell, and in this cell, this is a nucleus, and you can see these are the chromosomes. Chromosomes are present in the nucleus. So now I'll explain how the structure of chromosome will be. This is a cell. In the cell, nucleus is present, and you can see the thread-like structure here in the nucleus. This thread-like structure in the nucleus. These threads are called chromatids. They are usually tangled in this fashion when present in a regular active cell. When the cell is active, these chromatin fibers are present like this. They are entangled and freely present in the nucleoplasm. Is it possible to divide the single cell into two equal halves? These are in the form of tangles. It is possible to divide this into two cells? No, it is not possible. So imagine if we have these different color threads. Imagine if we have different reels of thread of different color, and if thread tangled together, if we were to separate them, then we should be first thing to do, we will first separate each thread, then make separate bunches. In a similar way, even the cells separate these chromatin threads. This process is aiding by the condensation of the thread. It means these chromatin materials will gradually start coiling like a telephone wire, and it will again gain a structure like this. This structure it will gain. It is present in a thread-like structure, tangled, and it is not possible to divide this tangled chromatids to divide into two cells. So for that, what does it do? It get coiled like a telephone wire. It gets coiled together and form a chromosome, and we can see this kind of structure only during the cell division. So that it becomes easier to divide these chromosomes into two daughter cells. So we are familiar with this kind of structure. This is the structure of chromosome. This is a chromosome. So you may ask, what are chromosomes? So we can say that both chromatids and chromosomes are the same. So these chromatids and these chromosomes both are same, but the name is different for the different structure. It is thread-like structure, so that's why it is called chromatids, and these threads get coiled and it forms chromosome. Now next question is, what are these threads or chromosome, to be precise, made up of? So what are the components of these chromatids or chromosomes? So till here we have understood. Now we'll see who discovered chromosome. Discovery of chromosome. Chromosome was first studied by a German scientist in the year 1882. German scientist Walther Fleming in the year 1882. He noticed that there is a rapid dividing cell of the larva of salamander. Salamander is an amphibian. So when he observed that under the microscope, he saw minute threads appeared to be dividing lengthwise. Fleming called their division mitosis, literally meaning thread. Next we will see the components of chromatin. It is made up of two substances, that is DNA and histone protein. DNA and histone protein. 40% DNA is present and 60% histone protein is present. Each chromosome contains one long DNA molecules associated with many proteins, that is histone protein. This complex of DNA and protein is called chromatin. A single large DNA molecule is very large, and hence it is described as micromolecule. DNA is called micromolecule. If you unwound all the DNA in a single cell, it would form a string that is around 2 meter long. If you unwound means you take out the DNA molecule from the chromosome, so if it will be approx 2 meter long. DNA is called deoxyribonucleic acid. DNA and this histone protein, these two together arranged, DNA is coiled with histone protein. How it is coiled, I'll explain. See, this is chromosome. If we take out the thread from, like how we take out the thread from the reel, it is arranged. So if we take out the thread from this, how it will look? It is arranged with the, see, this is histone protein, and this histone protein is wrapped with the thread. That thread is DNA. This is thread. This is histone protein, and this thread is DNA. Imagine I'm taking a rope and taking a ball. So if we are coiling the ball with the rope like that, the histone protein is like a ball and rope is a DNA thread. So we are coiling the ball with the thread like that. DNA is coiled with the histone protein. Each single DNA strand is composed of repeating nucleotide. Before that, I am going to explain some few terms. Kind of nucleic acid. First of all, I will explain kinds of nucleic acid. Kinds of nucleic acid. DNA, RNA. Second, components of chromatin. DNA, histone protein. So we have seen this DNA and histone, these are two together arranged. DNA is coiled with histone protein. These histones are organized to form a unit of 8 protein subunits to form a histone octamer. What is octamer? Means 8 histone protein will be present. See, octamer, 8 proteins will be present. 4 we can see here. We can see 8 histone protein is coiled with DNA. 8. See this 8 octamer will be arranged and coiled with this DNA, histone protein, and this is DNA. This is called the structural unit of chromosome or chromatin. This is called nucleosomes. What is nucleosomes? Nucleosomes is a structural unit of a eukaryotic chromosome consisting of a length of DNA coiled around a core of histone protein. This is histone protein and it is coiled like this. Again it will be coiled, and inside this 8 histone protein will be present. Each single DNA strand is composed of repeating nucleotide, which are made up of 3 components. Now, this DNA is made up of 3 components of nucleotide. First is pentose sugar. pentose sugar. Second is phosphate group. And third is nitrogenous base. See, this DNA is made up of these three things, that is pentose sugar, phosphate group and nitrogenous base. I'll explain the structure of DNA. Structure of DNA, it is double helical structure. Each strand of DNA is made up of repeating nucleotides. See, this is double stranded. You can see double stranded structure of DNA will be present. And each strand of DNA is made up of repeating nucleotide. Now, what is nucleotide? Nucleotide is the basic structural unit of DNA. Components of nucleotide is pentose sugar, phosphate group, nitrogenous base. This pentose sugar and phosphate group, they form the base of they form the strand of the DNA. And this nitrogenous base, they are the adenine, cytosine, thymine and guanine. They form a strand between this. This strand is made up of pentose sugar and phosphate group, and they are bound together. They form a ladder like structure. This is made by nitrogenous base. That nitrogenous base is of four types, that is adenine, guanine, thymine and cytosine. Adenine, cytosine, thymine and guanine. See, this is the basic structure of DNA. The strand of the DNA, if we see this strand, so how it will look? It is made up of phosphate and sugar, pentose sugar. They form a strand. Of DNA, and this base, the base will be adenine, guanine, cytosine and thymine. So these are the bases of DNA, nitrogenous base. So adenine will bound with thymine, there will be two hydrogen bond will be present between adenine and thymine, and in guanine and cytosine there will be a 3 hydrogen bond. So here it will form like this. So adenine with thymine, and there will be 2 hydrogen bond between this. Guanine, cytosine, there will be 3 hydrogen bond will be present in between. Like this, the arrangement will be like this. See, this is phosphate, sugar, base, and here this is a hydrogen bond between the two bases. Hydrogen bond between the two bases. Each single DNA strand is composed of repeating nucleotides. So this DNA is made up of repeating nucleotides, and these are made up of phosphate, sugar and base. 4 types of nitrogenous bases are present, that is adenine, guanine, cytosine and thymine. Adenine will pair with thymine with 2 hydrogen bond, and guanine will pair with the cytosine with the 3 hydrogen bond. Nitrogenous base. we categorize them into two, that is purines and pyrimidines. Purines, in this adenine will be there, adenine and guanine. Pyrimidines, thymine, cytosine and uracil. Thymine, cytosine, uracil. This uracil will be present in RNA. It will be present in RNA. In RNA, instead of thymine, uracil will be present. Instead of thymine, uracil will be present in this RNA. Four things will be present, that is adenine. Adenine will be present, guanine will be present, cytosine will be present, and uracil will be present. So the basic and structural functional unit of our DNA is nucleotide and chromosome is nucleosome. So this is the basic unit of DNA and unit of DNA. The shape of DNA is given by Rosalind Franklin, and the double helical model of DNA was given by Watson and Crick. The shape of DNA was given by Rosalind Franklin. The double helical model of DNA. What is double helical? This double stranded structure is called this. This is double helical structure. Was given by Watson and Crick. Was given by Watson and Crick. The shape of DNA molecule given by Rosalind Franklin in the year 1953, and this was given by the Watson and Crick, and they got the Nobel Prize in the year 1962. Got Nobel Prize in the year 1962. This phosphate backbone is a portion of DNA double helix that provides structural support to the molecules, a chemical compound made up of 1 phosphate and 4 oxygen atom. Now next is we will see what is gene. Genes are specific sequence of nucleotide on a chromosome that encode particular protein which are expressed in the form of some particular feature of the body. It's like किसी भी डीएनए में कुछ पोर्शन ऐसे होते हैं जो प्रोटीन के लिए कोड करते हैं, और कुछ ऐसे पोर्शन होते हैं जो किसी भी प्रोटीन के लिए कोड नहीं करते हैं, या ये बोल सकते हैं कि इस पोर्शन से कोई प्रोटीन नहीं बनेगा, देन दैट इज कॉल्ड जीन्स. तो डीएनए के वो पोर्शन जो प्रोटीन के लिए कोड करते हैं उन्हें कहते हैं जीन. ये प्रोटीन ही हमारे बॉडी में किसी न किसी कैरेक्टर को गवर्न करेंगे. एग्जाम्पल, मान लीजिए हमने पर्सन के डीएनए लिया और इस डीएनए के किसी सेगमेंट में आई कलर की जीन है. इस जीन को हम बोलते हैं बी जीन. ऐसे प्रोटीन के लिए कोड कर रही है जो ब्लू कलर का पिगमेंट प्रोड्यूस करती है. सो we can say that portion which encodes for particular protein, and that protein will code for some characteristics that is called gene. So this is gene. Genes are specific sequence of nucleotide on a chromosome that encode particular protein which are expressed in the form of some particular feature of the body. See, this is DNA, and this nitrogenous base in the DNA is adenine, thymine, guanine, cytosine. All this see, if this part of the DNA encodes some protein, and that protein may be like if it shows the characteristics of, like some person is having blue color eyes, or this part will show the color of the hair, it may be brown, black or something. So the particular sequence of this nucleotide on some chromosome will encode particular protein, and this helps in expression of some features on the body, that is called gene. Now next we'll see the structure of chromosome. So we have seen that the chromosomes get condensed during cell division. At the time of cell division, the chromosome will get condensed. Otherwise, when it is not dividing, at that time this chromosomes get decondensed, and it is like a thread like structure in the nucleoplasm. So when this chromosomes they form, so this, the upper part is called the telomere. These two, one and this is the second, these two are called sister chromatids. These two are called sister chromatids, like this. These two will be called sister chromatids. This is called centromere. Each chromosome contains hundreds and thousands of genes that are precisely coded for proteins in the cell. The centromere is called the primary constriction. This centromere is called primary constrictions. The kinetochore provides the center of polymerization of tubulin protein and assembly for microtubule. So during the, we will study about kinetochore in cell division. So these chromosomes are categorized into four types, that is metacentric, submetacentric, acrocentric and telocentric. Metacentric, submetacentric, acrocentric and telocentric. This metacentric, as we can see, the centromere is placed almost exactly at the center. Meta means between, so it is placed. Centromere, we can say, is almost at the center. It is present at the center. In submetacentric, the centromere is placed slightly away from the center. So it will be placed away from the center. This will be metacentric. So we are dividing all these four types of chromosomes according to the centromere where it is placed. In the submetacentric, it is slightly away from the center. So this is submetacentric. And the third is acrocentric. In this, the centromere is seen almost towards the end region. So in this is acrocentric, almost at the end of the present, almost at the end, towards the end region, that is acrocentric. Telocentric, this is telocentric. Centromere is present at the extreme end, and this kind of chromosome is not found in humans. So there are if you talk about in humans, there are 23 pairs of chromosomes are present in humans. So there are 22 are autosomes and one pair is sex chromosome. Upper part is called arm P and this is called arm Q. That's all for today. Thank you.